well, right? Always think about this in a positive sense. Always think about it in positive ways. The power of positive thinking. I haven't yet decided where we're going to make the break for the midterm. I will announce that next week. It'll probably be sometime on Monday, but I haven't decided where that's going to be yet, and I will let you know. I will also schedule a review session for the exam, and I'll announce that next week as well. Okay, so 2 things to look forward to. Oh, and one other thing. During the week of Thanksgiving, I may have mentioned this before, but I'll say it here, you're not required to go to recitation. So the TAs will be available at the normal recitation times from Monday through Wednesday. Obviously, they're not going to be there on Thursday. They will be available in their regular office hours in their offices at those times. So if you want to see them during your normal recitation time on Monday through Wednesday, you can do that. They'll be there. Okay? So that's one good piece of news, right? Okay, so we got pretty well through signaling last time, and I hope that what you started to take away in terms of your understanding of signaling is that this process is really critical. It's critical for the cell's ability to respond to its environment, and it's also critical for the cells making a decision about whether or not to divide. All right? Now, we're actually glossing over this in a very simple fashion. I know it seems like, oh my God, this is going to this, 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 but it's actually even quite a bit more complicated than what we're talking about. All right? But I try to keep it at a level that we can see the bigger picture. The bigger picture here are these controls. All right? The steps are important to some extent, but the bigger picture is what these controls are actually doing. So when I talked about the EGF receptor, you could see how that EGF receptor and EGF itself we're helping cells to make the decision to divide or not to divide. And we'll see other examples of this, one of which I'm going to talk about, in fact, 2 of which I'm going to talk about very briefly here, Sark and BCR-ABL, which I'll give some information about. And we can start to see how not only if we understand signaling, we can understand processes like cancer, but we can also begin to understand how we might have strategies for treating them. Okay, so these are really important considerations. There's also another oncogene that is relevant that I'm not going to talk about here. I'll talk about next term, and that is an oncogene called P53. It also plays a very important role in the decision of the cell to divide or not to divide, and in this overall signaling process, but it's more relevant for our purposes for what we'll talk about next term with respect to DNA replication. So we'll save that for that time. All right, well, right now I want to talk a little bit about Sark, SRC. SRC is another proto-oncogene, and I hope you remember that a proto-oncogene is the unmutated form of an oncogene. The proto-oncogene plays an important role, as does any proto-oncogene, in controlling cellular processes. Now, with respect to oncogenes themselves, they usually play roles in the processes relating to division or not division. All right? It's usually where proto-oncogenes are found. Okay? Sark is one of those. Sark has an interesting history. Sark was actually the first oncogene that was ever discovered. Right? And amazingly enough, it was discovered over 100 years ago. And it wasn't realized what it was at the time it was discovered. But what they knew was that chickens who got infected with a certain virus, called Rous sarcoma virus, right? Chickens that got infected by a certain, you don't need to know the name of that virus, by the way. Chicken that got infected by a certain virus developed cancer. And so this was really interesting, okay? The idea that perhaps cancer was infectious, A, would be of concern, and B, told us something about the process of cancer. Now, that Sark, the reason that this virus was cancerous, why it was causing cancer, was it turned out that the virus along the way had picked up a piece of a proto-oncogene. A piece, not the entire oncogene, but it picked up a piece of it. The piece that it picked up, as you will see, was what gave it the properties of causing a cell to divide uncontrollably. So in order to understand that piece, I need to tell you a little bit about what Sark does and why that piece was critical. Okay? So, in simple terms, what Sark does is when it is in the active form, it acts like a tyrosine protein kinase. You guys have heard this before. It acts like a tyrosine protein kinase. Microphone's acting up today. All right? And that tyrosine protein kinase can phosphorylate a variety of things on tyrosine residues and cause the cell to divide. So Sark is playing, again, a role in the decision to divide or not to divide. And I hope you're beginning to get the picture that there's a lot of things that can play roles in that decision. It's not one decision, but there's many things that can determine if a cell is going to divide or not divide. When Sark is activated, it causes the cell to divide. When Sark is inactive, it sits there and does nothing. So for many cells, Sark is sitting there doing nothing because it's in the inactive form. Well, what's the difference between the active form and the inactive form? Well, it turns out Sark is regulated as well. And Sark gets regulated by phosphorylation of tyrosines. This phosphorylation of tyrosines theme keeps coming up again and again and again. Now, this one's a little backwards to what you've seen before. Sark has a tail. That is, it has two ends. It has a tail. It has a carboxy tail. That is, the carboxyl end of Sark has several tyrosines in it. When those tyrosines get phosphorylated, Sark is turned off, not on. So Sark, when it has phosphates on its tyrosines, doesn't stimulate cell division. It's when those tyrosines are gone, when those phosphates are gone on those tyrosines, that Sark causes the cell to go on and divide. All right? So it's the absence of the phosphates on Sark that causes it to be active. So if I take Sark and I remove the phosphates off of its tyrosines, it's going to cause the cell to divide. You with me? All right. What did this virus do? It grabbed the piece of Sark that was missing the tail. Meaning it didn't have those tyrosines that could get phosphates on there that could turn Sark off. The piece of Sark that the virus picked up was only in the active form. It could only, when it infected a cell, cause a cell to divide, 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 divide. Now, I'd like you to think for a minute why that might be beneficial for a virus. Why might that be beneficial for a virus? What's that? Reproduction of the virus. When a cell is dividing, what's it doing? It's making copies of its DNA. What does a virus need to do? Make copies of its DNA. What does it take to make copies of its DNA? Plenty of nucleotides, right? This worked very well for the virus. Didn't work really well for the home organism, but most viruses don't work very well for the home organism. Instead of simply killing a cell and moving on to the next cell, like a cold virus might do, this guy was probably killing cells and also stimulating cells to divide and make more copies of the virus. Right? So, Sark is a good example of how a proto-oncogene might be altered and cause a cancer. Do we have Sark? Yes, we do. Might a mutation in Sark cause a cancer in a human? Yes. What would be the nature of that mutation? What's that? Loss of a tail would be one. Changing of tyrosines to something else that couldn't get that phosphate. Right? We can think of a variety of ways that we could alter Sark that might result in Sark being on. We see that on signal on when it shouldn't be on. We're potentially talking about uncontrollable growth. Yes, ma'am? That's correct. So the question is, I said Sark was active when it didn't have its phosphates, and her question is, how does it lose its phosphates? Okay, I'll ask you the question. I think you guys know the answer to this. What is the answer? How does something lose phosphates? Phosphatase is a great way to lose phosphates. Another great way would be not to have the phosphates put on in the first place. Maybe something that was controlling the kinase that phosphorylates Sark. If it were made inactively, that would leave Sark in the active form, right? So, it doesn't matter how that happens. What matters is if Sark is not phosphorylated, then it's going to stimulate cell division. And hey, we want cells to divide sometimes, right? What we want is controlled growth. The difference between controlled growth and uncontrolled growth is the difference between being you and me and being a cancer cell. Yes? Yes. Can Sark phosphorylate other things? Sark is a tyrosine protein kinase. So it's participating in phosphorylating other things that in turn ultimately make cells divide. So it's part of that signaling process that results in cellular division. Okay? Yes? Good question. Her question is, does Sark insert its mutated form of Sark into the cell DNA? It could, but for the most part it doesn't need to. For the most part it doesn't need to. Its mere presence will be enough to cause the cell to divide uncontrollably. And there are viruses that do insert things into that cellular genome. HIV is a really good example. Okay? HIV is a really good example. Okay, let's see. So that's Sark. There's the structure of Sark if you want to memorize something. Probably not. The last thing I want to talk about is another kind of tyrosine protein kinase. This one is interesting, and this one actually has a very cool and interesting organ connection. All right? Now, I need to tell you a little bit about this. There's a proto-oncogene called Abel. ABL. All right? ABL is this thing keeps cutting out. ABL is a tyrosine protein kinase. And you can imagine what it does. It's involved in that decision process about cells to divide or not to divide. Okay? There's a real pattern to these things. Abel is a proto-oncogene, and it helps the cell to make that decision. It does it by phosphorylating proteins that ultimately help make that decision for the cell to divide or not divide, right? When Abel is active, it causes cells to go through the process of division. All right? Now, in the normal scheme of things, Abel functions as we would expect it to function. Either on or off, divide, not divide, right? There is a recombination that can happen. Now, recombination involves taking pieces of DNA apart and putting them back together in different ways. Taking pieces of DNA apart and putting them back together in different ways. Imagine a puzzle that you make, that you put the puzzle back together and you don't put the pieces exactly where they go, but they fit. You get a picture that looks odd, right? You'd get a picture that looked very odd if you did that. Well, that's what's happening in recombination of DNA. Pieces that didn't start out next to each other can end up being next to each other. That's what recombination is all about. Okay? Well, there's a recombinational event that can occur in cells that takes a piece of chromosome 9 and links it to a piece of chromosome 22. And this happens at a low frequency, but it happens sometimes in cells. If that happens, you have problems. And one of the problems you have is that a piece of a gene called BCR, which is normally a gene that's expressed at reasonably high levels in cells, meaning that there's a fair amount of it made. Okay? Now, BCR, it turns out itself, is also a proto-oncogene, but that's not necessary for our purposes here. All that we care about with BCR is the fact that BCR is made in reasonably high levels inside of cells. In this recombination, the front part of BCR, which includes the control region that determines how much of it is made, the front end of BCR is linked to the tail end of ABL to create something we call BCR-ABL. Now, This creates a brand new protein, part of it from BCR, part of it from Abel. The part of it from Abel still has the tyrosine protein kinase activity. All right? What the BCR contributes is the fact that there's a lot of it made. You put these two guys together, and now you've got a brand new protein that didn't exist before, that's a hybrid of the two, that a lot of it is being made, and it's a tyrosine protein kinase. It's easy to regulate a small amount of Abel. It's not easy to regulate a large amount of BCR-ABL. What does that mean? It means if you make a lot of this thing, there's a pretty good chance some of it's going to be active that you don't want to be active. And when it's active, you got it. It's going to stimulate cell division. And when it stimulates cell division, that where cells shouldn't be dividing, you guys know that as uncontrolled growth, and you know that as cancer. This rearrangement that you see is a common rearrangement we find in a type of leukemia. Okay? A common rearrangement we see in a type of leukemia. It's been known for a long time what happens. It's been known how it happens and so forth. Now we get to the Oregon connection. Okay? There's a professor up at OHSU named Charles Drucker. Anybody know Charles Drucker? A couple hands. D-R-U-K-E-R. Okay? He's interested in things that we can use to treat cancer. And his idea is that, well, this is a different kind of protein. It's not the same as BCR. It's not the same as Abel. And it's a tyrosine protein kinase. What if we were to design and use an inhibitor that stopped the activity of tyrosine protein kinases? Might we be able to stop this cancer in its tracks? That's a good question. Knowledge about signaling pathways allows us to ask questions like that and address them, okay, with a therapy. Well, he gets knocking around and discovers, hey, there's a drug company that's got this perfect compound. It's called Gleevec. G-L-E-E-V-E-C. Gleevec was designed by a drug company, but they couldn't really see much of a market for it, so it's sitting around doing nothing. They don't figure they can make any money on it, and when they don't make any money on it, they publish what they've done with it, and it sits there. And he says, hey, this thing is a tyrosine protein kinase inhibitor. Let's look at what we know about signaling. Wouldn't it be really interesting to try this and see if, in fact, this knocks out this type of cancer? So he tries it. And it works. It works really well. He has to go knocking on a lot of doors to convince anybody that this thing's going to make any money. Tells you something about the drug industry, doesn't it? He spends a lot of time knocking on a lot of doors and finally gets enough things out there where there's a lot of clinical testing and a lot of positive results. And all of a sudden, oh yeah, maybe we could make some money on this thing. Okay? Gleevec is one of those drugs we think of as almost a miracle drug. It is very, very powerful at treating certain types of cancers. Okay? It works particularly well on this type of leukemia because this brand new protein that's made seems to preferentially bind Gleevec compared to other tyrosine protein kinases. That's kind of cool. Because when we think about that, we think about, do we want to inhibit every possible tyrosine protein kinase that we have? Might have some problems, right? If we can tailor it for a brand new protein that's not found anywhere else in the cell except for only in cancer cells, if we could find something that only knocked out this BCR-ABL and no other tyrosine protein kinase, we would have something called the perfect silver bullet. A silver bullet is something that kills only the thing you want it to kill and not other things. Okay? It's not a perfect silver bullet, but it's a very, very good one. It's saved the lives of thousands of people. It has saved the lives of thousands of people. The course of treatment is not overly invasive. It is not something that causes a lot of side effects. Okay? As a result of this, Dr. Drucker has, in fact, been discussed as a potential winner of the Nobel Prize in Medicine. It says something about what you use, what you learn. It says something about persistence, okay? And it says something about making a difference in the lives of people. That's really cool. That's what biochemistry is about. Okay. That's BCR-ABL. And that's Gleevec. Questions about that? Yes, sir? What makes BCR-ABL so prone to bind with Gleevec? Well, if you think about what we talked about with respect to protein structure, what's going to affect binding of a protein to a substrate? What's going to affect that? Structure, right? So it turns out that conveniently, this has a structure that has a great binding site for Gleevec. It has a great binding site for Gleevec. Gleevec was created by a drug company before that was even known, but that actually turns out to be a really, really good match. 3D structure is really critical. Would you say it has a high Km or a low Km for Gleevec? Has a low Km, right? Right. Very cool. His question is, were they able to predict what it was able to bind to because of its shape? Not really. He was interested in it because it was known to be a tyrosine protein kinase inhibitor. Yeah? Timeline. The question is, when was this? Sometime back in the 90s, but I don't know the exact date. Yeah. Dr. Drucker came and gave a seminar here at OSU last fall. Wonderful, wonderful seminar and a great informative man. Yes? Question, is it a reversible binding? Yes, it is. Yep. It is, yep. And people who take Gleevec take it for a fairly long period of time, actually. Yes? What? Why is it difficult to what? Why is it difficult to control this gene? Well, what you have here, the main reason this is a problem is because you have a high dose of the gene. ABL in small amounts is regulated by something else, but if you overwhelm that regulation by something else by making a lot of it, it becomes difficult to turn it off. We saw how turning it off was critical for Sark. If we overwhelm that regulatory scheme, the likelihood some of those are going to slip through and be active when we don't want them to be active is higher. That's what's happening here. All right, great. Well, that's what I want to say about signaling. I hope I've convinced you that signaling is a pretty cool process and that you can learn something really useful about how cells work by understanding signaling. We're getting ready to turn our attention now to how all of these things come together and regulate metabolism. Okay? Metabolism is the next phase of the course and the first half of BB451. Metabolism is what I like to think of as the chemistry that's going on inside of cells, more specifically, the biochemistry that's going on inside of cells. Metabolism is the sum of all of the biochemistry, all the biochemical reactions that are happening inside of cells, we call metabolism. Okay? Here's a schematic diagram you've probably seen in other classes, describing what's happening in the metabolism of glucose. We'll focus our early metabolism studies on glucose. Okay? Glucose is broken down in a pathway called glycolysis. That pathway involves a 10-step process whereby the 6-carbon glucose molecule is converted into 2 3-carbon molecules known as pyruvate. This process is energetically favorable. And energetically favorable processes will go forwards. Now we'll see that even though the process is favorable, there are some individual steps inside of glycolysis that have some barriers that cells have to get over. Some of these have barriers that cells have to get over. And by understanding energy, and understanding how energy affects reactions, we will better understand what those barriers are and how cells get past them. Okay? How cells get past them. All right. Well, in order to understand the barriers and understand the energy considerations, we obviously have to spend some time talking about Gibbs free energy. In your recitations, the TAs have been going through some problems with you with respect to Gibbs free energy, and now we're going to address some of those concerns and some of those thoughts about how free energy affects directions of reaction. Okay? Eventually, you're going to know this. Isn't that kind of scary? Yeah. Not in this term. Okay? Not in this term. In this term, we're going to basically be working from about right here down to about, from about right here down to about right here. Okay? This is this little blue thing we're going to see, we're going to work on. All right? Now, when you look at this, the first thing that I hope pops into your head is that this certainly looks like a road map. Perhaps a bit demented kind of a road map, but it might not be unlike something we might see in a major metropolitan city. That road map includes a loop. It actually includes two loops. There's another loop that is right over here. Okay? All right? But for the most part, we see highways leading in. We see streets branching off. We see stopping points, kind of like subway stations. And understanding what each of these different points is helps us to better understand the city. We jump on the light rail. We know all the listings of the stops on the light rail, right? And each of these stops on a metabolic map corresponds to a specific molecule. Okay? The other thing we see about this metabolic map that I have in front of you is that the metabolic map is very interconnected, just like a city street map is very interconnected. If I want to go from this point here down to this point here, there's many ways I can get there. Now, I tell you that because when we get studying metabolism, we're going to largely be doing this with blinders on. You need to recognize that that metabolic pathways aren't the same as the way that we teach them and the way that you're going to learn them. What does that mean? We're going to focus, as I said, from right here down to right here. And we're going to talk about going from right here down to right here as if all these other side streets don't exist. But in reality, they do. This molecule right here can go down. This molecule here can go to the left and then up. It can go down to the left and then right. We see there's a lot of different directions things can go. So that means that when we try to think of a pathway, which is what we see here, we like to think of it as if there are no branches to it. That this molecule is always going to go to this molecule, is always going to go to this molecule, is always going to go to this molecule. But in fact, in the cell, this molecule at the top is in a soup. There's nothing that makes it go to the next one, to the next one, to the next one. It's going to go where it's most likely to be used. That's something to keep in mind, right? And you'll see this becomes more of a consideration the further we get along in metabolism. But we will teach it as if we're going from here to here to here to here to here, and nothing else existed there. Everybody with me? Okay. So, now, I said I'd talk about energy. Let's talk about energy. ATP, as I've referred to in class, is the gasoline of the cell. It is the primary, not the only, but it is the primary energy source of the cell. Other nucleotides are also used for energy. ATP is used in a very wide spectrum of reactions across the cell. It's by far the dominant energy form in the cell. The making of ATP by the cell is a very critical process. If we stop the ability of a cell to make ATP, we kill the cell. Okay? ATP is the triphosphate form. You know that ADP is the diphosphate form. AMP is the monophosphate form. And yes, AMP is different from cyclic AMP. Okay? The form with the most energy is the ATP. And the reason it's the form with the most energy is these phosphates really don't like each other. Negatives repel, they are repelling each other, and the repulsive energy between those phosphates that's there is stored. That's stored chemical energy. When we break the bond and we release a phosphate, we are releasing that chemical energy. ADP, therefore, has less energy than ATP does, and AMP has the least energy of the three. There's no repulsive phosphate there. Okay. Let's now turn our attention to the delta G equation, which you've been seeing in recitation. Okay? Delta G is known as the change in Gibbs free energy, and in simple terms, this is what the delta G equation looks like. Delta G is equal to a term called delta G0 prime plus RT times the natural log, the concentration of products over reactants. That's actually the simplification I made. What really matters for our purposes is the value of that ratio. And this ratio and this equation looks very, very much like the Henderson-Hasselbalch equation, which had pH, which was a variable, equals pKa, which is a constant, plus the log concentration of salt over acid, right? Delta G is a variable. Delta G0 prime is a constant for a given reaction. RT is a constant. And natural log is just a different log term. It has the same rules as the other log term does. If the concentration of products over reactants is greater than 1, the value of this will be negative. If the concentration of products, will be positive. If the concentration of products over reactants is less than 1, the value of this term will be negative. It doesn't say what the value of delta G will be, because delta G is equal to the sum of this plus this. Right? So the log term, for example, could be negative, but if the delta G0 prime term is more positive, then the overall delta G will be positive, right? Thus, it's possible to have more reactants than products, but still have a positive delta G. Make sense? Just like you had to do in the Henderson-Hasselbalch equation, where you had to be able to recognize how the value of the products over reactants changed and how that effect that had on the, in the Henderson-Hasselbalch, where the value of salt over acid changed and the effect that had on pH, similarly, you're doing the same thing here. How does the value of the products over reactants change, and what effect does that have on the delta G? Now, let's talk about what delta G is and what delta G0 prime is. They're really similar. Their names are similar, and students frequently confuse the two because they sound similar. They're not the same thing. Okay? What is the delta G? The delta G is the change in Gibbs free energy for a reaction. You learned in freshman chemistry that the value of delta G tells us the direction of a reaction. If the delta G for a reaction is negative, the reaction moves forward as written. If the delta G for a reaction is positive, it moves backwards as written. And if the delta G for a reaction is 0, the reaction is at equilibrium. As I've noted previously in class, equilibrium does not mean equal concentrations of everything. Okay? It simply means that the concentration is not changing over time. That might mean that you've got 10 times as much products to reactants, but once it's reached that stable state where it's not changing, it's at equilibrium. Okay? Let's think about what this means for what we've just learned. If I told you I had a reaction at equilibrium, and the delta G for the reaction was, the delta G0 prime for a reaction was positive, what would you tell me about the value of products over reactants? It would tell us that the natural log term has got to be negative Because at equilibrium, everything's got to be equal to 0 for delta G. So if I have a positive of one, I have to have an equal value of negative for the other. What does it take to have a negative value for the log term? The ratio less than 1, meaning more reactants than products. Okay? What if I told you I had a reaction in which I had equal amounts of reactants and products? What would you tell me? If the ratio is equal to 1, that is reactants equal products, what's the natural log of 1? 0. What's the value of the log term? 0. What's the value of delta G? Delta G 0 prime, right? Now, this gives us a good definition for delta G 0 prime. Under standard conditions, we measure standard conditions at a fixed temperature, a fixed pressure, and 1 molar products and reactants. Everybody's equal. Delta G is equal to the delta G 0 prime under standard conditions. So we can simply say that delta G 0 prime is what the change in free energy is, only under standard conditions. Only under standard conditions. The delta G is a much more valuable term for us than delta G 0 prime because it tells us the direction of a reaction under any conditions. Right? What if I told you that the delta G 0 prime for a reaction is negative, and I had equal concentrations of products and reactants? Which direction is the reaction going to go? It's going to go forwards, right? That turns out to be really useful because when we talk about individual reactions, and we'll be talking about these in all of metabolism, I'm going to give you many scenarios just like that. I'm going to give you a delta G 0 prime value. I'm going to say this is very positive. And when I say that the delta G 0 prime value is very positive, I think that what should pop into your head is, if I start with equal amounts, that sucker's going to go backwards. And if the delta G 0 prime value is very negative, or negative at all, and I start with equal amounts of reactants and products, it's going to go forwards. Okay? So it gives me a really good idea about what is happening inside the cell. It tells me what I can think of as a general tendency for a reaction. It doesn't tell me the absolute direction of a reaction. Why not? What's that? No, it's not anything to do with rate. It's a trick. It's not a trick question, but it's a tough question. Okay? It tells us only the direction of a reaction when we start with equal amounts. In cells, we don't always have equal amounts. So it's a general tendency, but the actual direction will depend on the actual concentration of products and reactants. But it's very useful for thinking of the tendency to go forwards, or the tendency to go backwards, or the tendency to go both ways. What if I told you the delta G for a reaction was equal to 0? There's a good question for an exam. What would that tell you? It would actually tell you something I've already talked about. It would tell you that equilibrium would mean equal concentrations of products and reactants. That's the only time that would hold. The only time that would hold. Okay, well, let's look at an example. Let's think about this in a practical term. The example I'd like to talk about is something people can relate to with their bodies. It's actually this reaction right here. Okay? This is a reaction that occurs in your muscles. Okay? Creatine is something that people who think they are athletic, or people who think that they want to do something with their body, think is really great for them to take. Because creatine is a compound in our muscles that actually helps us to store energy. I'm going to tell you how it works, and then I'm going to tell you some dumb things people do with it. All right? Look at this reaction. This reaction says creatine plus ATP goes to creatine phosphate plus ADP. We're using ATP, we're making creatine phosphate, and look at the delta G 0 prime. What does the delta G 0 prime tell us? Here. Does this reaction go very far forwards, or does this reaction tend to go backwards? It tends to go backwards. If I start with equal amounts of everything, it's going to go backwards. Right? It turns out, let's imagine that I am sitting around like you guys are. I know you're using a lot of brain power right now, but you're not using a lot of muscle power. You're sitting here, your muscles aren't burning ATP. Right? Unless you're punching your neighbor. All right? Your muscles aren't using ATP. What's the concentration of ATP in your muscles? It's high. When you're not using ATP, the concentration of ATP in your muscles will be high. ATP is one of the reactants. All right? As I increase the amount of reactants, which direction will the reaction tend to go? Forwards. Le Chatelier's principle, right? I disturb an equilibrium, I add a lot of this, it's going to push it to the right. So when you're sitting here, your muscles have a fair amount of creatine phosphate. It's overcoming that delta G0 prime by what? By having a high concentration of reactant. You can show that in the delta G equation. All right, the fire alarm goes off, and you see a fire blow up here on stage, and you start running out of here. You start burning ATP, right? Your ATP concentrations go down, right? What happens when your ATP concentrations go down? Well, let's think of two things. You're thinking about the reaction. One thing is if your concentration of ATP goes down, what happens to your muscles? Right? You've got to get way out of here. You've got to get far away. And if you don't have something to back up that ATP, what happens to the ATP? Your ATP falls, your muscles don't work. Right? This guy's a backup. When ATP concentrations start falling, what happens to this equation? It starts moving to the left. And when it starts moving to the left, what does it make? ATP. So even when the muscle's concentration of ATP starts to fall, you've got something that starts replenishing it. That's really cool. And it's available on demand. These things happen instantaneously. That's really great. Okay. Now, people say, oh, well, then if I want a lot of muscle power, then I want to have a lot of creatine phosphate. Because the more of this have, the more capacity I have, and the better off I am. So it's a real popular thing to go out, and gobble up creatine and so on and so forth. Dumb idea, in my opinion. All right? But I want to give you one case where it's a totally stupid idea. And you can understand it by understanding this equation. I want to go run a 100-meter dash. I want my muscle energy to be high. I want to make as much of this stuff as I can because I want that to kick in. When I am going, I want that ATP doing me a favor. I'm going to go have a whole bunch of creatine. What's going to happen to my ATP concentrations if I make a whole lot of creatine? I make a lot of creatine phosphate. High concentration of creatine is going to do what to the reverse reaction? Is the reverse reaction going to go as much? No. What's going to happen to my running ability? Look stupid? So then I got these brainstorms that say, well, what if I go ahead and load a creatine phosphate? Okay? Creatine phosphate's really great inside your muscle cells. Eating creatine phosphate isn't going to get it into your muscle cells. Now, there are reports and there are claims that this will increase your muscle and your energetic capacity. My concern is this is messing with a very fundamental aspect of cellular energy. Not a good idea. Why isn't it a good idea? Let's think back to ATCase. ATCase was activated by what? ATP, right? And what was ATCase doing? It was making plenty of pyrimidine nucleotides in preparation for what? Cell division. We start messing with cellular energy. What do you suppose we may be messing with respect to cellular division? Maybe not a good idea, right? Okay. Now, I know of no studies that show an increase of cancer or any of these things with it. But if it were me, I wouldn't be messing with something like that. Okay? I wouldn't be messing with it. Okay. Enough hammering on the head. Any questions about that? Do you understand what's happening with respect to direction of reaction and the concentration of creatine phosphate? You could do this on an exam. You could make this argument on an exam. And everybody's getting a sort of a wavy look in their face, right? Should we nail it with a song? Oh, question, okay.
So adding creatine would make up ATP creatine phosphate, but wouldn't it just get released again?
So adding creatine would do what now? It would make it go this direction, but if you have a lot of this, it's not going to go away, and so you try to make this reaction reverse, and one of your products is going to be high. That reaction is not going to go backwards very well. Okay. Yes, another question. Shh, 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 shh.
You're saying that
Shh, shh, shh. I can't hear.
it's not very favorable reaction. But does that mean that it's not very favorable to go right?
So the question is, does it mean when I say a reaction is not very favorable, does it mean to go to the right or to the left? It means in the direction I'm talking about. So it's not very favorably going to the right if I start with equal concentrations of the two, because I would have a positive delta G value. That's not going to go to the right. Come see me. I'll explain that to you. Okay. So here's something about muscle energy. It's called the Muscle Energy Song. And I can't sing it, but David Simmons can sing it well. You guys can sing along. Changing words.
Steady as ever when whatever energies arise, the action is exacting, believing in the end. My pride grows all contracted, using energy extracted from reactions that react in me, using ATP.
All right.